Hey folks, Professor Finn here. Today we're going to study another chapter in pathology and microbiology for funeral science students, in this case, chapter 22. Like, share, subscribe, get this to a friend, get this to your parents, share it with your pets, fall asleep at night listening to it. That's a little awkward. Probably don't do that. That freaks me out a bit, but you get the picture. Let's get this stuff out there. And let's get up the screen and talk about chapter 22, the physiology of bacteria. Regardless of size, every life form has certain requirements that must be met to maintain life. They all have food, oxygen, moisture, temperature, pH, light, and osmotic pressure requirements. A large part of controlling the spread of disease is understanding the survival requirements of different microorganisms and then changing the microorganism's environment so that these requirements are not met. No requirements, dead microorganism. Death and destruction of certain microbes by the body allows them to release injurious poison. So even when we kill them, they can still hurt us. First up, nutrients. Different microorganisms have different food requirements. Some microorganisms are capable of meeting their nutrient requirements without an external nutrient source. Others require nutrients from specific types of food sources. Some microorganisms are capable of adjusting to the changes in the availability of external nutrient sources in their environment. They can support their growth by feeding on alternate internal nutrient sources when an external nutrient source is unavailable. Like when you, as a student, have to go grab that cup of noodles because leftovers are all gone. And this is why you typically go visit your families to stock up the fridge, right? At least that's what I did, and I'm unapologetic about that. Autotrophic bacteria. Autotrophic bacteria are self-nourishing bacteria capable of growing in the absence of organic compounds. Autotrophic organisms are capable of obtaining nutritional value from the carbon and carbon dioxide. One of the largest groups of autotrophic bacteria are cyanobacteria, which derive their name from their appearance, which is similar to blue-green algae. They are not, however, algae. Their colors may differ, as the Red Sea is named as such because of red-colored cyanobacteria. Your book succinctly states that the scum that exists near the edges of ponds is the result of autotrophic bacteria, and that it can cause disease in humans if ingested. And this leads to the question, who the heck wants to eat pond scum? I'm telling you, I've never walked by a pond and gone, gotta get me a bowl of that. Ugh. Autotrophic bacteria produce about 20% of the Earth's oxygen and are capable of converting inorganic carbon dioxide into the nutrients they need through photosynthesis. Autotrophic bacteria use light and carbon dioxide to produce food just like plants. Cyanobacteria are responsible for the continuation of the nitrogen cycle because they can convert atmospheric nitrogen into its organic form, which most life forms in the world require for survival. Autotrophic bacteria do not cause disease in humans, but they do cause some human diseases like swimmer's itch. They also participate in the decomposition of human remains. Heterotrophic bacteria require complex organic food from a carbon source to grow and develop. Heterotrophic bacteria are the cause of numerous diseases in humans. Some heterotrophs are able to survive in a wide variety of organic compounds, and your book gives a good example. Pseudomonas is capable of consuming naphthalene, which is an active ingredient in mothballs and also a paint remover. And if you tried to consume naphthalene, you would get a quick trip to the ER. Not all heterotrophic bacteria cause disease. These types of bacteria are used during the process of fermentation, which produces adult beverages and other necessary foods for one of my favorite holidays, which is Oktoberfest. There are three categories of heterotrophic bacteria: strict or obligate saprophytes, strict or obligate parasites, and facultative bacteria. And it is these little buggers, the facultative ones, that everyone gets angry about. Take a breath, it's really not that hard. So, obligate saprophyte. A strict saprophyte or an obligate saprophyte are organisms that can only survive on dead or decaying matter. A strict parasite is completely dependent on their living host for the nutrients they need to survive. All viruses, Viroids and prions are obligate parasites because they lack the internal structures to produce energy or utilize food sources. Facultative bacteria. Facultative bacteria can adapt to a differing source of nutrition. As food sources in the environment change, facultative bacteria simply feed on the new food sources or produce their own nutritional sources. Oxygen. All organisms have oxygen requirements. The requirements for microorganisms range of those those who cannot survive if any oxygen is present, to those that must have an oxygen-rich environment. There are 5 classifications of bacteria based on their oxygen requirements: strict or obligate aerobes, strict or obligate anaerobes, microaerophilic organisms, facultative organisms, and aerotolerant organisms. First up, obligate aerobes. An obligate aerobe is a microorganism that can only live in the presence of oxygen because they need oxygen to metabolize sugars. If these aerobes do not have oxygen, they are unable to produce energy. Some intestinal diseases are caused by obligate aerobes. Bacillus and Pseudomonas are typically strict aerobes. And if you see something like that, that's probably something you should jot down and memorize now, because it's going to be brought up when we talk about Bacillus and Pseudomonas later on. Obligate anaerobes are the exact opposite of the ones we just talked about. They are microbes that can only survive in an environment devoid of oxygen. Even compounds containing oxygen, such as hydrogen peroxide, can be deadly to such microorganisms. Listeria, Clostridium botulinum, Clostridium perfringens are all obligate anaerobes. Not all bacteria have such requirements of all or nothing when it comes to oxygen, and this is what upsets everyone in the world. So first up, we have microaerophiles. Microaerophilic microorganisms require little free oxygen, 2 to 10%. That means these microorganisms need to have a stable environment with no more than 2 to 10%. If it's below 1%, they die. If it's higher than 10, they die. Microaerophiles, 2 to 10%, okay with oxygen at that percentage level. Facultative bacteria are capable of adjusting to changes in oxygen levels in their environment. Facultative bacteria may care about how much oxygen is in the environment, how much oxygen comes and goes, but it's not a deal breaker in how they live their life. Aerotolerant organisms can grow in the presence or absence of oxygen. So there's a key word that triggers aerotolerant. Aerotolerant can grow in the presence of the absence of oxygen. So you don't have to go too terribly crazy trying to figure out what's the difference between microaerophilic and aerotolerant and all. Aerotolerant can grow in the presence of or absence of oxygen. These differ from facultative organisms because they don't care about whether oxygen is in the environment. Okay, the word you're focusing on for aerotolerant is the word grow. Streptococcus pyogenes is an aerotolerant bacteria that causes a variety of bad things, which we will study in a future chapter. All microorganisms require moisture, but the amount necessary for growth varies between species. Each microorganism has a maximum, optimum, and minimum amount of water necessary to sustain life. Generally, bacteria that can cause disease in humans require fairly high levels of moisture to grow, while molds and yeast that can cause human diseases can grow in lower moisture levels. Wording is hard today. This is why most bacterial diseases occur in the body's tissues, while most fungal diseases occur with greater frequency on the surface of the body. Temperature is a factor in the growth of microorganisms. There are 3 broad categories of bacteria based on their temperature requirements for growth: psychrophiles, mesophiles, and thermophiles. Organisms do not necessarily fit perfectly into each of these ranges, as some bacteria may grow at the extreme ends of each range. The organisms may also grow a little faster or a little slower based on the temperature. Each organism has its own minimum, maximum, and optimum temperature for growth. The minimum temperature is the temperature low, which bacterial growth will not take place, while the maximum temperature is a temperature above which bacterial growth will not take place. We're going to talk about death times and all that stuff, later. But the minimum temperature is the temperature low where bacteria simply will not grow. They will hibernate, they will do something, but they will not spread. And the same is true for the maximum temperature. Once it crosses that threshold, they also will not grow. May not kill them, but they will not grow. The optimum temperature is the temperature at which the bacteria grow best. Okay? Psychrophiles are bacteria that prefer cold, thriving at temperatures between 0 and 25 degrees Celsius, or 32 to 77 degrees Fahrenheit. Mesophiles are bacteria that prefer moderate temperatures and grow best between 25 Celsius and 40 Celsius, or 77 degrees to 104 degrees Fahrenheit. Thermophiles are bacteria that grow best at high temperatures between 40 degrees Celsius and 70 degrees Celsius, or 104 degrees to 158 degrees. Some bacteria grow in extremely high temperatures found inside volcanoes at 105 degrees Celsius to 110 degrees Celsius, or 212 to 230 degrees. Mind you, water boils. If you're trying to kill stuff, you want to boil it at 100 degrees Celsius for 10 minutes, and yet we're seeing here that there's some bacteria which grow at that temperature. So by boiling it, you're actually just giving it a nice warm bath. These variations in temperature preferences explain why some bacteria only grow in certain parts of the body. They may even impact how a disease is treated. So, for instance, a treatment for syphilis used to be giving the patient malaria, so that the fevers associated with malaria would kill the bacteria that cause syphilis. Thank God medical science has progressed slightly since then. Variations in temperature preferences are important to us as funeral professionals because changes in body temperature occur before and after death. An antemortem fever would likely result in agonal fever as well as the opposite. The body may be cooler than normal antemortem, which result in agonal algor. The natural cooling of human remains occurs at the average rate of 4 degrees the first hour after death and 1 degree Fahrenheit each hour thereafter until room temperature is met. Super important thing. The cooling of human remains. In the first hour, you drop 4 degrees. And then every hour after that, you drop 1 degree until room temperature is met. Be very, very, very careful, because you may get asked a question about what is a body's temperature after X number of hours, and that is lower than room temp, and the body can never be lower than room temp. Okay, so what do I mean by that? Let's say your body is at 99 degrees. Okay, the body is at 99 degrees. And the temp outside is 93 degrees. And what they say is, this person passes away, body's 99 degrees, the room temperature is 93 degrees, and the body sits in this area for 10 hours. What would be the body's temperature at the 10th hour? And what you might be doing in a rush is you might be going 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. In the first hour, I lose 4, then I lose 1, and 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 1. Then we're going to break that down over here, right? That's 2, 2, 2, 2, 4. And then we have 4, and we have 4, and 4, and 1. And now we have 13 degrees down. And if you're like, Professor Finn, did you really have to write all that out? No, I didn't, but it illustrates something important. Don't be afraid to use that whiteboard on your national board exam or that scratch paper for your test to write something out, slow yourself down, and check your math, okay? So if my body over 10 hours has dropped 13 degrees, I might say that the body is going to be 86 degrees, and you would be wrong, because the body stopped cooling any further once it hit 93. And I promise you, if they're asking a question like this, you will have 86 degrees as one of the answers, absolutely. And you might see that, run the math, and jump right to it, and you lose. Don't take the bait. Placing the deceased in a refrigeration unit reduces bacterial decomposition by slowing or retarding the growth of mesophilic bacteria. We're never freezing the body. This would result in embalming complications, but we are dropping the temperature so low that most bacteria do not grow, and it also slows down the autolytic processes by which the body would be decomposing. Sorensen scale, or pH scale, is used to indicate the concentration of hydrogen ions present. The pH ranges from 0 to 14. 0 is the most acidic, 14 is the most basic, and neutral is 7. A human body is a pH of 7.4. Super important. You are slightly alkaline, okay? You are slightly alkaline. If you've not listened to the embalming lectures already, when we get into the embalming lectures, I promise you, you're going to hear body is slightly alkaline a lot. Critters that can hurt us generally check in at this pH level. Go figure. If this is where we are at and stuff hurts us, that pH can't kill them. Okay? Note, the external pH will not outright kill some microorganisms, and they will keep right on trucking as long as the pH level of their cytoplasm does not change. We need to get inside the cell, mess with the pH level inside the microorganism in order to kill it. Just because the pH on the outside has changed, they may have something like a capsule, or they may have mucous membranes or something to prevent the environment from hurting them. You need to get inside and shock them. Helicobacter pylori can survive stomach acids because they contain an enzyme that allows them to split the urea found in the stomach into ammonia and carbon dioxide. Yeast and molds can also survive well in an acidic environment. Light. Light also affects microbial growth, depending on the type of microbe. The type of light may also matter, as your book references some bacteria can be destroyed by ultraviolet light, and we'll talk more about that when we're talking about controlling microorganisms. Osmotic pressure. Osmotic pressure is the pressure required to prevent the net flow of water across a semi-permeable membrane, such as the cell membrane found in bacteria. Translated into layman's terms, the pressure on the liquid outside the cell equals the pressure of the liquid on the inside of the cell. Pressure, in this case, has nothing to do with the concentration of solutes. Pressure, in this case, has to do with the concentration of solutes in the solvent that is the liquid both inside and outside the cell. So what are they talking about? Osmotic pressure means that there is something in a liquid, okay? Flavor aid, sugar, whatever it's going to be. And when we put a membrane next to this and another liquid, the liquids are going to want to react until some level of osmotic pressure is attained where it's equal. Otherwise, it's just going to continue to flow and flow and flow and flow and flow and cause problems. When the fluids outside of the cell contain high concentrations of salt, the water inside the bacterium is drawn into the external fluid. This condition results in the cell membrane being drawn away from the rigid cell wall, and the cell membrane then collapses upon itself, effectively killing the bacteria. So what's happening? Okay, when the fluids outside of the cell, draw yourself a little diagram. When the fluids outside of the cell contain high concentrations of salt, okay, we're just going to put, here's our salt. What is going to happen? All the H2O that is in here and the small amount of H2O that is out here, this water is going to want to go out there and try to dilute it. That is the osmotic pressure. The water is going to flow from inside the cell to the outside of the cell. And if it happens rapid enough, what's going to happen is the cell is going to collapse upon itself. And that's what kills the microorganism. One, all the water goes out, and then basically the skin falls in on itself. As the salt concentration outside of the bacterial cell increases, the bacterium attempts to compensate by producing certain amino acids that help maintain the necessary water levels inside the bacterial cell. Small changes in osmotic pressure may be compensated for by the bacterium but the larger changes result in its death. So it knows something is happening, and so it tries to take some mitigation to prevent that water from leaving. So if it's a slight change, it can balance itself out. But if it's a drastic change, it can't do it. It gets shocked and it dies. As a general rule, molds and yeasts are capable of withstanding greater changes in osmotic moderate pressure than other microorganisms. Basically, molds and yeasts are uber against all others when it comes to these things. The use of salts as a method of preserving meat, such as pork and fish, has occurred throughout humankind's history because the high concentration of salt retards microbial growth and inhibits the composition of proteins. Osmotic pressure is high in many embalming fluids, which is one of the reasons that it retards microbial decomposition and decreases the spread of infectious disease from the deceased to the public. Most microorganisms do not grow in isolation. The different types of associations microorganisms form with other organisms are related to both the formation of disease in humans and the decomposition of human remains. Although the types of associations are important, it is equally important to recognize that in the real world it is not always possible to distinguish the level of benefit or harm any organism receives from its associations. There are 5 forms of symbiosis: mutualism, commensalism, parasitism, synergism, and antagonism. These are definitions you memorize. Okay? Symbiosis is a form of association when 2 or more different species of organisms live together in close association. Period. End of story. Symbiosis: 2 or more different species of organisms live together in a closed space. Doesn't say one is better, one is living off the other. Period. They're living in close proximity. End of story. Mutualism occurs when 2 species live in close association and both benefit. Okay? Mutualism. They live in close proximity and both of them get an advantage from this. Escherichia coli exist in the human digestive tract, where they produce vitamin K and certain vitamin B vitamins. They also permit the breakdown of cellulose, which would otherwise be undigestible. Neither E. coli or the human is harmed in this situation, right? E. coli gets fed, they give us something, and we benefit from them being in our guts. They get outside of the guts, and we have all sorts of problems. One small thing changes, and E. coli messes you up. Commensalism. Commensalism occurs when one organism gains some benefit, but the host is not harmed. Okay? With mutualism, both organisms, the host and the microorganism, get a benefit. With commensalism, only one gets a benefit, but the other isn't harmed. There are numerous bacteria that live on the skin of human beings, and they serve no purpose, but neither do they mess with you at all. Your book refers to remoras and sharks. Win for the remora. Parasitism. Parasitism is just like commensalism, except the host is harmed. So I don't know why they say it's just like commensalism. Parasitism means you're living in close proximity, one is getting a benefit, and one is being injured. All living organisms can act as a host for parasites. Typically, the host is a macroscopic organism, while the parasites are microscopic organisms. Occasionally, some parasites are larger multicellular organisms like roundworms and flatworms. Synergism. Synergism occurs when the harmonious action of 2 microorganisms produces an effect that neither could produce alone. So this is important. Okay, this is where people really get messed up with this. Okay? Synergism isn't they're both receiving benefits. Synergism is saying that when they are together, their actions produces an effect neither could produce alone. Your book references synergistic infections due to animal bites, especially human bites, which can permanently destroy tissue. While the bacteria present normally in the mouth are not individually harmful, okay, I lick someone's skin, which is absolutely disgusting. None Nothing happens other than probably you're going to get arrested for doing something stupid in public, okay? When they're forced into the tissues you bite by the crushing power of the jaws, you develop a serious infection. So normally, bacteria don't do anything. The moment you put them into this new environment, they then can cause a problem. So synergism does not necessarily mean good or bad. In this case, it definitely means bad. It simply says that when they are put together, now you have a problem. They produce an effect that neither could do by themselves. Certain enzymes are released from some bacteria that increase the rate of growth of other bacteria, and aerobic bacteria are utilizing available oxygen so that anaerobic bacteria can grow where they normally could not. So again, bacteria do something, it then removes oxygen from the environment, and then these other little buggers can grow where normally they couldn't. Again, that is a synergistic relationship. They are producing something that normally neither could do by themselves. This creates a perfect storm which results in irreversible tissue damage which any of the involved bacterial species could not have caused by their lonesome. Antagonism is a mutual opposition, contrary action, and in the case of microbial relationships, the term refers to the inhibition of one microorganism's growth by the presence of another. So antagonism isn't like parasitism where a host is harmed and something gets a benefit. Antagonism refers to a microorganism inhibiting another microorganism by their simple presence. That's how you keep that one straight. The microbiota of the normal flora of the body, the bacteria that are always present but yet are not causing any harm or disease to the human host. Antagonism occurs when the normal flora inhibit the growth of other microorganisms by competing for nutrients or space or by producing toxins that kill the microorganism. Your book references lactobacilli in the human vagina that reduces pH levels to such an acid environment that other microorganisms are frequently incapable of growth, thus reducing the risk of vaginal infections. Folks, that concludes this lecture. We'll see you next video. Once again, like, share, subscribe. Drop something in the comments. Let me know if you found this useful or where you're from. And